far we've looked at some slides that we've made ourselves using the wet mount process. And I did leave one thing off of this. Make sure that you dry the excess liquid off of the slide before you put it under the microscope, and that's to protect these lenses from water and chemicals like methylene blue that could damage the lens. So we have made wet mount slides of an onion, a green plant called Elodea, and then we use the methylene blue to dye our own cheek cells and look at those. But we can also look at preserved organisms, and we're going to look at a few of these today. The organisms we're looking at are protists, and protists are eukaryotic cells, which means they have a nucleus in them. Now, there are 4 kingdoms of eukarya. You could have plants, animals, fungi, or protists. And protists are unique because they don't fit in with the other kingdoms of eukarya, but they may have some characteristics that they share between 2 or more of these kingdoms. So we're going to be looking at some slides today called Euglena, and you can see when you have a preserved slide that the name is going to be written on the slide for you. We're going to be looking at Paramecium, so you can see where that's going to be written on the slide. And notice that you can't see anything under these slides yet. We are going to have to get them under the microscope because these are unicellular organisms. We're also going to be looking at amoeba, and so these are the 3 big protists that you need to know for biology, but we're also going to look at one more called Volvox. And you'll notice that Volvox, you can see little purple specks in that slide because Volvox is a multicellular protist, whereas most protists are going to be unicellular. So Volvox is a little bit bigger. So I'm going to put each one of these slides under the microscope by putting the slide on the stage. Making sure that I put the slide under the stage clips, and then we're going to focus in the same way that we did before. We start on our lowest power. We have the objective far away from the stage. As we look through the eyepiece of the microscope, we're going to use the coarse knob to lower that objective toward the stage until we see detail on what's on the slide. And of course, once we get that detail, we switch to the next highest power and make sure that you center the organism that you want to look at before you switch powers. Once you get to the medium power, we're going to use the fine focus to toggle back and forth to get different amounts of detail. And then once you're satisfied with that, you can go to the high power and again, use your fine focus to toggle back and forth to see different levels or different layers of the organism, or to see better detail. So let's go put these under the microscope. So now let's look at the protists that we were able to see. We started with our amoeba, and this is our amoeba on low power. They have all of these unique and crazy shapes. As we increase the power of magnification, you can see more detail. And once we're on the highest level of magnification that we have, which is 430 times, you're able to see the nucleus really well, and these extensions of the cytoplasm. And these are called pseudopods, and that's how an amoeba is going to move. It stretches out this pseudopod and latches onto a surface, and then the other side of the amoeba is going to let go of whatever it's holding on to, and it's going to push its cytoplasm toward that pseudopod. And pseudopods are also able to engulf food for the protist. These are Paramecium on low power, and if we increase the power of magnification, they're going to get bigger. And once we get on high power, you can see this nucleus in the cell. You can also see this fuzzy outline of the Paramecium, and that indicates the cilia that are present on the outside of the Paramecium. And these cilia are tiny little hairs that are going to allow the Paramecium to swim through its environment. And they can also sweep food into the gullet of the Paramecium, which is kind of like the mouth for the Paramecium. So if we look at the Euglena on low power, you see that they appear red, but Euglena are really green. These Euglena lost their chlorophyll during the preservation process, so they have a pink color, but keep in mind, Euglena are photosynthetic, and so they are usually going to be green. As we increase our level of magnification, we see these Euglena get bigger and bigger, but they are the smallest protists that we're looking at. You can see that they have a nucleus, and what you can't see is that on this non-pointy end, they do have a flagella, and that flagella is a long tail that spins like a propeller and helps these Euglena to swim through their environment. We also looked at Volvox, which is our only multicellular protist. And this is the Volvox on low power. As we increase power, you can see more and more detail. So this is high power, and each one of these little purple dots here is an individual cell, and they all work together to make this colony. Now, these dark spots that you see inside are not nuclei because this is not an individual cell. These are more colonies of Volvox that are going to mature inside of that organism and then break free to go forth and start their own colony. So far, all of the cells that we've looked at under the microscope have belonged to domain Eukarya, which means all of these cells have a common characteristic. They have a nucleus and other organelles within their cell membrane. So we have 4 kingdoms of eukarya, and we have looked at 3 of those kingdoms. The only one we haven't looked at is fungi, but we have looked at plant cells with our onion and our elodea, and we noticed that plants are always multicellular. We have compared those plant cells to animal cells by looking at our own cheek cells, and we know that we are also multicellular. We got lots of cells out of the inside of our mouth. And so we've also looked at the kingdom Protista, and protists are eukaryotes that do not fit into the other 3 kingdoms. They might have characteristics that might place them in both, and so they are given their own kingdom of Protista. And those organisms can be unicellular, like we saw with our Euglena, our Paramecium, and our Amoeba, or you can have multicellular protists like our Volvox. So now we are going to see if we can find some living protists in this tank of water that's been sitting here over the weekend. So it's got a little bit of slimy stuff that looks like it's growing on the top. So I definitely want to get some of that slimy stuff in my pipette, and I want to go around at the bottom and get a little bit of what I see growing at the bottom and maybe even in these rocks. So I'm going to just try to get a little bit of water from all over. And I'm going to use a slide to make a wet mount of what I'm looking at. And I might make 2 or 3 wet mount slides to see if I can find some different living organisms moving around under my microscope. So I'm just going to take my liquid this time, which has the specimen in it, and I'm going to take my cover slip and place it on at an angle. And I'm going to put this under my microscope. I'm going to draw off the top a little bit, and I'll take some videos and pictures of what I'm able to find, and we'll talk about those in your lab. Here you're able to see some of the protists that we found in the aquarium under medium power. I don't know what all of these are, but we have here an example of a Paramecium. Looks like we have another example of a Euglena here. So we'll look at these on higher powers as well. So here are some of the creatures that you were able to see under the microscope. These very teeny tiny organisms are bacteria. They do not have a nucleus inside of them. You can see there's no spot in there. But all of these other organisms are larger, and they are eukaryotic, so they do have a nucleus. And you can see one of those nuclei right here in this cell. This is an example of a nematode that I was able to find, and you can see it moving around and munching on some of this algae. And that you can actually watch those food particles pass through the intestines of this nematode or tiny little worm. So we have a few post-lab questions here. What is a protist? And keep in mind that protists are eukaryotes. They have a nucleus, but these organisms do not fall into the other 3 kingdoms of eukarya. They may have characteristics that might put them in 1 or more groups, so they get their own group called protista. The structures that these protists are going to use to move, the amoeba use the pseudopods, the extensions of those cytoplasms. The Euglena used a flagella, which is the long tail that helps it to swim. And the Paramecium uses cilia, which allows it to swim through its environment. And these are short hairs, and there's lots of them on the outside of a cell. Paramecium and amoeba can also use their movement structures, their pseudopods, and their cilia in order to get food. The only multicellular protist that we looked at was Volvox. But keep in mind that when we looked at the protist videos, that all of these little organisms moving around under your slide were in a single drop of water. So you can imagine how many of these single-celled organisms are going to be in a pond. And if you drink pond water, then you are probably going to be drinking some of these protists. And these protists can then set up infections in your body that can be quite dangerous, like Giardia, for example. Make sure you fill out the pre and post lab questions for your lab and submit that to Canvas so that you get credit for this assignment.